Can you guys hear me? Good. So I'm Seth Walk. I'm in the Department of Microbiology and Immunology. So I'm going to talk to you today. Wow, this is really loud. Turn it down just a kind of a, I get to yelling when I talk. So, so I'm going to tell you a little bit about a project that we've done in the lab in wastewater with the causative agent of COVID-19, which is SARS-CoV-2. And because this is a biotech class, I wanted to put in some technology that we're using to do this. So you can go to the next slide. So we're going to go through a little bit about DNA, how we look at this molecule in the context of SARS-CoV-2 diagnosis, and then something we call surveillance. And that's just looking for this organism out in nature or in people or in animals. So we're going to talk a little bit about genomes and how we look at them. And then SARS-CoV-2 in wastewater, I'll tell you a little bit about the work we've done here in Gallatin County and how this same procedure or process is being used around the country to look for COVID-19. Okay, so genomes, we all have them. All living cells have to replicate themselves and pass on their genetic information to the next generation. And that's how evolution happens on Earth, right? So our cells have genomes, but all microbes, so bacteria, archaea, they all have genomes. Even viruses have genomes. And if you take a philosophy of science class, they'll probably talk to you about whether viruses are actually alive or not. I can give you my opinion later, but anything that replicates itself tends to have a genome. All living things on Earth have genomes. Okay, so genome is just all the genetic material in a cell. Okay, so it comes in many different forms. A lot of us think of genome, we think of DNA, but there's actually, as I'll show you, RNA-encoded viruses. And your genome, you tend to think is in your chromosomes, right? But bacteria, for example, have a chromosome, and then they have parts of their genome that are called extrachromosomal. They're plasmids. They're things that, they're viruses. They carry them along as part of their complete genetic makeup, but they're not actually in their chromosome. Okay, so a genome is a much broader term, all the genetic information. In our cells, right, what parts of our genome are not encoded in our chromosome? Does anybody know? Has anybody heard of a mitochondria? So mitochondria are in each and every cell. There's many mitochondria in our cells, and they have DNA. And you get your mitochondrial DNA from your mom. That's not transmitted from your dad. So your mitochondrial DNA is sort of extrachromosomal. Okay, anyway, you can see that genomes also vary in size. This table lists in size, KB means kilobases, so those are 1,000 base pairs or 1,000 nucleotides, A's, T's, C's, and G's. And you can see that humans are about 3 million kilobases, so 3 million times 1,000. That's how big our genome is. And you can see that microbial genomes like E. coli is much smaller, and even viruses much smaller yet. Okay, so genomes come in all varieties, shapes, sizes. There's linear chromosomes like ours. There's circular chromosomes. A lot of bacteria and archaea have those. And so shapes, sizes, and molecule. So DNA encodes genomes, but also RNA. Okay, so there's a lot of diversity here. You can go on. Next slide. Okay, so how do we look at genomes? This is a great place to be, Montana State, because we're sort of right next door to the world's discovery of genetic material. Okay, and that might sound a little dramatic, but it's actually really true. So this guy, Thomas Brock, was from Indiana University, and he came out to Yellowstone to study thermophilic bacteria. Okay, so things that live in really hot environments. And he was working in Mushroom Spring. He isolated an organism, and he began to look at the enzyme that that organism used to replicate its genome or replicate its DNA molecule. And because of that discovery, others picked up on this and developed a technique that basically every molecular biology lab uses in the world. And that's called polymerase chain reaction, or PCR. Okay, and that wouldn't have been possible without this basic discovery by Thomas Brock right out in Yellowstone. Okay, so the cool thing here is that you take DNA, the molecule, and it's basically the same molecule in all cells. Okay, so you can get our DNA, DNA from an E. coli, DNA from a virus, and you can heat it up in those double strands that make that molecule split open. Okay, now if you cool the molecule back down, those base pairs will find themselves and they'll make that double strand again. Okay, but that's really a cool thing about the molecule. You can heat it up, cool it down, it separates, it comes back together. Now, what this reaction does, heats it up, and we put little primers in there. These primers identify one specific location in that whole genome, and they just sit there. And they sit there because when they anneal, you can see the temperature range there. It's like 48 to 72 degrees Celsius. Okay, so we're starting to cool it back down, but we haven't cooled it down completely so those two strands find each other. We're just annealing those primers on. And then we heat it back up a little bit to about 70 degrees. And this molecule from the bacteria Thermus aquaticus that Thomas Brock identified, that's when it's active, right? Because that's where Thermus aquaticus lives. It lives in this hot spring that's about 70 degrees C. So because of that, if you put that enzyme in there, it'll find those primers, the ends of those molecules, and it'll make new DNA. And it'll do that on human DNA, it'll do it on viral DNA, it'll do it on any DNA molecule on Earth. So you run this reaction once, and you start with one copy, and then after the reaction, you get two. And if you do that same reaction again, you'll split two of the copies apart instead of one, and you'll make those. So you go from 2 to 4, 4 to 16, so on and so forth. And that's really important because a lot of the biochemistry, a lot of the biotechnology involved with DNA simply is based on having enough to work with. Okay, we're not very good at doing anything with one DNA molecule, but when we replicate that molecule and make a bunch of it, then we can manipulate it, and we can do things to it, like sequence it. Or as I'll show you, we can quantify it. You can go to the next slide. Okay, so all of this has to do with what we call the central dogma of molecular biology. Okay, and this is basically how these molecules are made. So DNA replicates itself. That has to be true, right? Because that's how we pass on our genes to the next generation. We replicate ourselves. Bacteria do this, archaea do this, viruses do this. So that's that first sort of circle up at the top with DNA. It replicates itself. And that's what PCR does. It just replicates DNA. Now, sometimes when we want to make a protein, right, we have to take the information in the DNA and get it into amino acid sequences, and that's called a protein. Okay, so how does that happen? Well, DNA encodes information, and we have enzymes that look at that information and make RNA. RNA is a very closely related molecule to DNA, but it's a little bit different, and it encodes the information from DNA and lets another molecule in our cells read the RNA and make amino acid sequences. Does anybody know what molecule that is? what machine that makes protein out of RNA? Those are called ribosomes. Okay, so ribosomes are a machine. They're a complex of proteins and ribosomal RNA, and they come together and they form this little machine in our cells. And they read RNA and they say, okay, what amino acid does that encode for? And it grabs that amino acid, it brings it in, and attaches the next amino acid, the next. And these polypeptide chains, these mini stretches of amino acids, those are protein. So we can go from DNA, make more DNA, we can take DNA, make RNA, and we can take that RNA through ribosome and make protein. And that's what our cells do. Now those red arrows up there are processes that we know to exist, but they don't exist very commonly. And certainly our cells don't do this. Our cells don't have the equipment and the machinery to make RNA from RNA, to replicate RNA. Viruses do. Also, our cells don't have the ability to take RNA and say, I wonder what DNA that might come from. And can I make that DNA? That's called reverse transcription. Because that first process there from DNA to RNA, that's called transcription. We probably heard of that maybe in high school biology. But the reverse of that would be reverse transcription. And we don't do that, but viruses do. So those are special reactions. Now, this central dogma you'll learn a lot more about if you take biochemistry, if you take general microbiology, other classes on campus. If you're interested in biology, you will see these again, and you'll learn a lot more about them. But I just bring them up now because PCR takes advantage of that first DNA replication step and really gets us over the threshold of being able to work with this molecule. The other thing that's going to be important for today is that reverse transcription, because SARS-CoV-2 is an RNA-encoded virus. And we have to make that back into DNA in order to run PCR on it, in order to amplify the genome, okay? Next slide. Okay, so the genome is a single-stranded, or SS, RNA. You can see there it's in the box. So it's a single-stranded RNA molecule that the virus actually carries around with it looking for a new host. And it's what they call a positive sense. So RNA, if it's double-stranded, has a positive and a negative sense strand, and it only carries one of those, and the one strand that it carries is the positive sense. So that's just to let you know that we're talking about RNA when we talk about the SARS-CoV-2 genome. Next slide. Okay, so we're going to use this special one on the right to reverse transcribe it to DNA, and then we're going to replicate that DNA a lot of times, and we're going to quantify how much is there. Okay, next slide. Okay, so where does this all come from? Well, when SARS-CoV-2 or COVID-19 was first observed in China, folks got to working on this really quickly because they wanted to know how can we detect it. So in our country, this was done at the Centers for Disease Control and Prevention, the CDC in Atlanta, Georgia. And their scientists down there cracked the code of SARS-CoV-2 genome, and they made primers that we need to do this PCR reaction. And they started distributing those across the United States to the state diagnostic laboratories. All states have state diagnostic labs. Ours is in Helena. Most are in the state capitals, but if you live in a big state, there's probably multiple state diagnostic labs. So this is the what we call the assay, or the protocol that the United States began to use way back in February to identify and diagnose SARS-CoV-2. Okay, next slide. Okay, so a little bit more about this reaction. I said that, you know, the first time that you run the PCR reaction, you start with one copy, you double that, and then you double that, and then you double that. So this equation of doubling, right, is very interesting, but it leads to predictive value, okay? Because What happens is if we have how many PCR cycles here on this graph on the bottom, and we get a signal, and we can say, that PCR is happening. We're amplifying the DNA. We can detect it. Then we can use that number to sort of back calculate how many molecules must have been in my sample in order to see it at that cycle. Okay, I'm going to pause there to make sure we're all on the same page. So let's say I have one molecule, right? And I know that with my piece of equipment, I can only detect, the first time that I can detect DNA is when I have, say, 10,000 molecules. Then I can just watch and wait until I get detection signal. I know that that's 10,000 molecules, and I can use that equation of doubling to back calculate to say how many molecules must have been in my original sample. Does that make kind of at least a little bit of sense? Okay, so we call that number at which we can detect the reaction taking place as this threshold. And because we're working with cycles, it's called a cycle threshold, or CT. Okay, and that's not to confuse you, but if you've heard of CTs, then that should kind of make sense on this graph. Okay, next slide. So the cool thing is we can go in with a known molecule. So say our target that we want to look at is SARS-CoV-2. We can make a little chunk of SARS-CoV-2 genome, and we can use that as a positive control, and we can really get our equation down. That's called a standard curve. And this is just showing you sort of results for a standard curve. So if you look at this 10 to the 5th, that's how many molecules. 1 times 10 to the 5th is about 10,000 molecules, right? So if I had 10,000 molecules of that DNA in my original sample, that CT would come up to about 10. If you can't see this in the back, it's about 10 cycles in order to see it. Now, if I dilute that signal and say I only have 10 to the 4th, it'll come up a couple cycles later. Okay, so the higher the CT, the fewer numbers of that DNA that was in my original sample. Okay, so it's an inverse relationship, but this gives you a sense of where do those molecules come up? And at least in this, by the time you get down to 10 molecules, just 10 molecules in the sample, you're looking at a cycle threshold of about 26 or so. Okay, next slide. Okay, so coronaviruses, I said they're RNA encoded viruses. So this is typically what happens in our cells. These viruses are out there, they're being transmitted. When they find a host cell, they can't just go in, okay? They have to find the receptor on that cell. And for SARS-CoV-2, we'll come back to it and go through why that receptor is even more critically important. Once it finds its receptor, it gets internalized. It goes through its genome replication. It uses, remember, we don't have any enzyme in our cells that can make RNA from RNA, but the virus does, and it carries its own molecule. It encodes for it. So it replicates its own genome inside our cells, and then transcribes a bunch, well, not really a bunch, 11 different genes into proteins. Okay, some of those proteins labeled here, N, M, E, and S, are involved in just making more of itself. They're structural proteins. They need to make more of itself. And our cells help that. Viruses sort of take over our cells to do their bidding, and they make more of themselves with our machines. So our ribosomes, our endoplasmic reticulum, our Golgi bodies, these are all co-opted by the viruses just to make more of themselves. Once they do make more of themselves, then they get released and they go look for more cells. Okay, so when you are infected and you sneeze on someone, those viruses have to get to the right cells in our bodies, have to find the right receptor, have to get internalized, have to make more of themselves, have to get out of that cell in order to go and find a new cell. Okay, next slide. So where do these things come from? There are reports, the first one in the US coming from an animal was in a New York zoo from a tiger, probably not this tiger, but most of the coronaviruses that we know of don't infect humans, or at least don't give humans symptoms. But we know that a lot of animals carry them, either symptomatically or asymptomatically. And the diversity of these viruses in animals is much, much broader than the coronaviruses that we tend to see in people. And so that's how we know kind of where they come from. Because when we look at animals, we see them, okay? And a lot of those coronaviruses, of all the animals that carry coronaviruses, bats tend to carry the ones that are very closely related to the ones that infect humans. For example, SARS-CoV-2 genome was sequenced and compared with coronavirus sequences from a lot of different animals, and it was found, they found this one in a bat. This is a horseshoe bat, I think it's called intermediate horseshoe bat. It was like 96.2% identical to SARS-CoV-2 from people. Okay, so we can look at the similarity of these viruses to try to figure out where they come from. Bats probably play a pretty big role, but the reason I put up a tiger was the USDA now post reports, and they require vets to report when animals are infected with SARS-CoV-2. And in the US here, the USDA posts a table, and a lot of animals on there are domestic animals like cats. So by far, the most common animal in our country that carries the same virus that will infect us comes from domestic cats. So there might be a cat link here, but probably it was first introduced into humans from bats. Okay, so a little bit more about the cellular receptor. So the receptor on our cells, and this is getting into why and how we can look for SARS-CoV-2 in wastewater. Okay, so if you've not been paying attention so far, listen up. So SARS-CoV-2 uses this ACE2 receptor, and it's not really important what ACE2 normally does. For this talk, it's just important to know that the spike protein, so you can see the spike, the green little thing that is a structural protein that the virus makes, that spike protein's on the outside of the virus. And you can see that up top, those little green things on the outside. They find ACE2 on the outside of our cells, okay? And that's how the virus is first internalized. And it was found with sequencing that in comparing a bunch of different coronaviruses that SARS-CoV-2 is actually probably mutated its spike protein to identify and become very specific to our ACE2 receptor. Okay, so it's much more likely to hit our ACE2 receptor than it would be, say, a cow or a giraffe or a bat. Okay, next slide. Now, you can go to the next slide. Okay, so there's a database that you can look up. Where is ACE2 expressed in our bodies? Okay, and that's called the Protein Atlas. This atlas was put together from a bunch of different medical sources, sudden death victims, healthy folks that just got some analysis done. This is a plot of a woman and a man and where ACE2 is normally expressed in our bodies, okay? And that's shown in red. Does anybody, you don't have to be a scientist to recognize that there's not much going on in the lungs of these folks. There's not much going on in the sinus of these folks. Where are most of the infections? Why is it called SARS? What does SARS stand for? Severe, anybody heard this? Severe acute respiratory virus, or sorry, syndrome. Yeah, if I could spell. SARS, severe acute respiratory syndrome. So when you get sick with SARS, do you have cramps in your GI tract? Well, that's maybe, but it's not going to kill you from infecting your GI tract. Where does it kill you? In your lungs. right? That's why people get on respirators in the hospital. But look at the expression of that receptor in our bodies, much more highly expressed in the gut than it ever is in the respiratory tract. So what does that tell us? Well, it tells us that people that have SARS-CoV-2 are likely to have cells that line their GI tract infected with the virus, and that virus is likely replicating and being shed in the person's feces, okay? So that's why this whole thing can be done and why surveillance of wastewater makes a little bit of sense. Because we can detect the virus from people that are shedding it normally in their GI tract. Okay, next. So this just gives you an indication. You don't even see lung on here because it wasn't really even detected in this assay relative to other parts of our body. Okay, so by far the biggest bars here are in your small intestine, your duodenum. There's significant expression in the colon as well. Okay, next slide. Okay, so our lab has been involved with the county, Gallatin City County Department of Health, to do surveillance of wastewater. And this was a need that came on, I would say, in March and April. We started doing these, started looking in wastewater in June. And these data here represent some of the data that we've generated in 2 towns in Gallatin County. I'll point out why I put these up here in a minute. But if you're a health officer in a county like ours, you want to go out and look where disease is happening, the cases of disease where they're popping up, and you want to make sure that you identify those people and their close contacts and try to quarantine them so they're not infecting everybody else, you know, try to stop the outbreak from progressing. So when you have cases in a town, that's kind of easy to do because these folks are going to the hospital. With something like SARS-CoV-2, where the person may or may not be symptomatic, you don't really know who to test in a new community. And a county like ours, where we're right next to Yellowstone, we get all these visitors, so they're not really residents, they're just passing through. How do we know whether or not there's folks bringing the virus into those towns? It's not an easy thing to track. So that's some of the reasons why we wanted to look in the wastewater. So up top is Big Sky, Montana. And Big Sky is a very, tourist intensive town, but mainly in ski season. Once the summer comes, things kind of calm down. And you can see by May, when we first started sampling up there, we couldn't even detect SARS-CoV-2. It wasn't there. Wastewater consistently came back without any signal. And then around, I don't know, first part of June, we started getting a little bit of signal in our qPCR. We're like, wow, that's different. We never got that before. And then the next week, say right around the start of, or the middle of June, it really went up. And that told us, wow, here's a community that was negative, 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 and then boom, positive. So onto that same plot, we put, and you can see these on the left side of the graph, the number of cases. So we plotted those, when those cases arose in that community, onto the wastewater data, and we had sort of the aha moment where, yeah, there was like 1 or 2 cases, and then boom, there was this cluster of cases. And those clusters of cases corresponded exactly when we detected this large upswing in the wastewater, okay? And that's important because that told us that our assay that we were doing, the qPCR, was at least somewhat sensitive to when new cases arise in a community. All right? Now, that's why I put that graph up there. Take a look down at the other graph. This is in West Yellowstone. This community is right beside Yellowstone Park. A lot of the folks that come into Yellowstone from the West Gate, they go right through West Yellowstone. Okay, and so when we started sampling down there at the end of May, we found the virus. Found it right away in the first sample. We're like, wow, that's interesting, because it's such a small community. There was something there's only like 900 hookups in the whole town that went into the wastewater stream that we were sampling from. That's not a lot of people, but we found it. And then we plotted on the number of cases, and wow, yeah, there's been cases going on in West Yellowstone long before we even started the wastewater surveillance. So that again gave us some confidence that the signal in the wastewater is indicative of cases in the community, but differently, as opposed to Big Sky, where it's sort of, we've had these cases come in often enough where we're always detecting the virus. When we did the lockdown, right, the governor said, hey, we need to lock down, everybody go home. And the industries and restaurants and businesses in West Yellowstone shut down. Everybody started, well, talking about wearing masks and social distancing. There's a big gap in the cases. So we didn't really have any cases in West Yellowstone for a little while. And interestingly enough, the levels in the wastewater disappeared. Okay, so that graph told us, hey, when the cases go away, so does the signal in the wastewater. So the combination of these two graphs really should illustrate the power of looking for SARS-CoV-2 in wastewater. One, it's a great indication of when the virus is present, and the other, it's a great indication of when it goes away. Now, we do have information on how much virus is there, and that's sort of the relative abundance of the virus. The problem with interpreting those data from our graphs is that we only sample these communities once a week, okay? And we don't really have to do something like, okay, if I look at the level of virus, can I predict how many people in that community are sick? We would have to have daily data. We'd have to have a lot more information. We just don't have that. So we're not really able to say with much confidence, hey, the virus is at this level, that means this many people are sick. It'd be nice to have maybe in certain situations, but our data are really good at saying, is it there or is it not there? That's a good indication of whether there are or not new cases. Okay, next slide. Okay, so we're reporting this on our website, and this is a resource that the county health officer uses to predict where they need to go in and do more testing, or maybe where they don't need to focus their testing efforts, because all efforts during a pandemic have a finite amount, right? There's only so many people that can go and do contact tracing. So where do you want to send those folks? Do you want to send them to Three Forks or Big Sky or West Yellowstone? Well, maybe let's look at the wastewater data and see where more effort is needed. And then they're available for the community. It's a public site. Anybody can go and ask, if I live in West Yellowstone, what's the virus looking like this week? And you can click on these links. These link to PDF reports. And in those reports, we put the results, the raw data from that qPCR reaction that I was talking about. And those are the CTs. And based on our standard curve, we can report back also how many genomes were in a liter of that wastewater. Okay, so we use that equation that I talked about, doubling to back calculate how many viruses were there. So we usually do, this is West Yellowstone, so we usually do, let's see, we'd have 2 samples there, so we usually do things in replicates as scientists. It gives us some confidence on what we're actually trying to estimate. Okay, next slide. So how is this being used around the country? Well, a lot of folks are interested in wastewater surveillance for the same reasons that I talked about. But how is it put into practice? How does it help us during the pandemic? And this was just a beautiful illustration of how we might think about using this in more real-time situations. And this was at the University of Arizona. This was just a few days ago. This article came out in the Washington Post, and basically what they did at the U of A, as they call it, they hooked up these samplers, the wastewater samplers, at dormitories, and there's something like 21 dorms on their campus, and they were monitoring these every other day or every day, I forget, and they would look for positivity. And all of a sudden, one of these dorms lit up. And so they said, okay, we have a positive signal. Let's go into the dorm and let's test everybody. So they tested all 311 residents, and they found 2 students that were asymptomatic but positive for SARS-CoV-2. So before any outbreak in that dorm, before anybody even know they had it, they identified 2 people that were shedding the virus. They went in, they got those 2 folks, they identified them, and they quarantined them before they had a chance to spread the virus and make others around them sick. Okay, so would this have turned into an outbreak at the U of A? Nobody ever know that, right? We have no idea what our interventions do, because we don't do those controlled experiments where we just say, hey, this dorm's positive, let's just let it go. That'd be unethical, right? Because there's a clear and present danger if there indeed would be an outbreak. But this is how it could have stopped an outbreak that was about to occur. So you might be asking, why don't we do this on our campus, right? You go to the next slide. Well, this is our testing facility. This is for symptomatic students. I don't know if you've seen this on the website or in the media, but if you have symptoms, you go and get tested. Okay, and this was done just last week that we got this facility up and running, that we got the personnel out there to man it, to do the testing. But we started with symptomatic folks because these resources are very limiting. Having the right people in the right spot, having a spot to do this, getting the swabs, getting the tubes, getting the transport media, everything that is required takes a lot of logistics and a lot of planning and a lot of money. Okay, so this is just for symptomatic folks. Doing what we're talking about is for sort of asymptomatic or surveillance. But the bigger reason why we're not doing on a dorm level or on a building level wastewater surveillance is what happens after we detect a positive. After we detect a positive, we'd have to sample all of the individuals that are in those locations, right? So 311 at U of A had to be tested. And that's the real capacity that's a shortfall here locally and in our state. Go to the next slide. My lab has turned, we've pivoted away from wastewater surveillance. We still are involved in it. We collaborate with another scientist that's taken that over. But my lab has switched completely now to helping the state with our capacity for testing. Okay, and these are swab tests that you get when you go in for any diagnostic for SARS-CoV-2, whether it be in a hospital, a drive-up clinic, or symptomatic student tent over by the stadium, you'll get a swab. The swab will be shoved up your nose. It'll be very uncomfortable, but it'll let us know whether you carry the virus or not. So we've helped out the state by getting, we have about 10 people in my lab now that all they do is test for COVID on swabs. We have a lot of robots that help us move the liquid around, set up the PCR reactions, and then report back the results. There on the bottom right is one of our typical work days. We work in the biosafety cabinets. We wear these things called PAPRs, because if you ever wear an N95 mask, they're kind of rough on your face if you're wearing them for 10, 12 hours a day. These PAPRs are like little helmets they fit on. They're much more comfortable. So we're in there, we're looking like aliens, and we're doing a lot of testing. Right now, we're testing between 500 and 700 samples a day that get sent to us from Helena, and we report those back in a 24-hour period. So we operate from Tuesday to Saturday. Sunday and today we'll be kind of cleaning the lab up, getting ready for the next week's samples. So as we increase capacity for our testing, we might be able to bring wastewater surveillance online for the campus. And once we identify a positive dorm, then we can go in and test everybody in the dorm, maybe in our lab right here. And that's about all I have. I don't know how we're doing on time, but I'd be happy to take questions if if you guys have it. Yeah. Let me run over here. Say that again. It's a great question. So how does the flow rate of the wastewater system or the size of the pipe? It turns out wastewater systems are really different, right? How they treat water is based on the size of the community and the need. And so flow rates, right? You can imagine that when you flush the toilet, it goes down a pipe somewhere along the line. That pipe meets another pipe. They join the flow, so on and so forth, till it gets into the plant, right? So where you are at in the system has a lot to do with how much flow there is and the volumes. So what we use is called an auto sampler. And an auto sampler is just a piece of equipment that's put in line or has a tube that's put into the pipe, and it can measure the flow rate, okay? And it pulls off liquid at set intervals over a 24-hour period based on the flow rate. So the more the flow, the smaller the volume that it'll pull, such that over a 24-hour period, what we sample here, say in Bozeman, we can directly compare that to what maybe West Yellowstone or a smaller community might have. But that's a great question, because how do you compare these very, very different wastewater systems? And that has a lot to do with it. Other questions? Yeah. This is why it's in a gym, right? I gotta work out. I get to stick a fly rod in somebody's face. All right. This is a first for me. What do you got?
Are the same resources being used for testing the waste sewage system being used for personal tests? Like, is that the same resources that's now being split between the two?
Right. So resources meaning money, or do you mean like the physical?
Like on the news in the past couple months, you've been seeing a lot of stuff about, oh, they have to sort of, split between all these different places the same tests that they have to do. Is that like the same sort of thing? Or is it like different kinds of tests?
Right. So that's true. Like in the media, you'll hear a lot about testing and how there's limited resources, and we're able to do this many tests this month, but only that many last month. And yeah, so the resources, get a little feedback. So the resources involved in human symptomatic testing and for wastewater surveillance comes from the CARES Act from the federal government. The CARES Act is the coronavirus relief funds that get distributed to all the states in the United States. And some of these monies go to local municipalities. Those local municipalities then get to do that, spend that money how they see fit. Some of the communities like Big Sky, West Yellowstone, Three Forks, and here in Bozeman decided, hey, we want to know whether our community has the virus. So they put that money towards wastewater surveillance. Symptomatic tests come more directly from the government, from the federal government and the state government. And those resources they're a little bit different pool of money, but ultimately they all come from the federal government's allocation for testing. Importantly though, when they talk about infrastructure and capacity for coronavirus testing, they're almost exclusively talking about symptomatic cases. Asymptomatic surveillance, they usually don't talk about, and prioritization is important. They prioritize samples from symptomatic folks first.
Flu season is coming up. Are you thinking about also assaying for standard flu?
So that's a great question too. You can imagine when flu hits, you'll be symptomatic. You'll have the same symptoms as SARS-CoV-2 or COVID-19. And you want to know, do I have COVID or do I have the flu? So CDC, that designed the primers to detect SARS-CoV-2, also has an assay to split them and differentiate them apart. So the cool thing is about this qPCR, those curves are generated by fluorescence. Okay, and that fluorescence, we can make different colors. We can actually detect different targets in the same reaction. One has, say, a green color, one has a red. And there's an assay that we can use in the same sample and the same PCR that will detect both SARS-CoV-2 and flu. Now, hopefully, the people that carry both are very, very small number. It does happen, but it's rare. So hopefully, if when we get that assay up and running, which we are, we can differentiate whether you have the flu or SARS-CoV-2. Flu season starts in October, so we have a little bit of time, but everything seems to be rush in this pandemic. Other questions?
You have a very active lab. I've suggested to the class that if they're interested in this kind of work, they email you and possibly interview with you about working in your lab.
Yeah, thanks, Dave. We have a lot of undergrads in our lab. Typically, we have, I don't know, at any one time, 3 to 5 folks working in the lab. We study the gut microbiome, and that's how I got interested in the wastewater surveillance and SARS-CoV-2 in the gut. So we study the microbes that live in and on our bodies that help keep us safe, keep us healthy, and then the ones that also cause disease. And then the other part of our lab now is the swab testing, the surveillance, the testing capacity. And undergrads are getting signed up to work in this lab as well. Unfortunately, there's only parts of the process that you can actually do in this kind of lab because you have to have a lot of training to work with infectious diseases. But it's a great way to get experience. If you're interested in the biomedical research field or you want to become a doctor or nurse, it's great experience to get involved in that. So yeah, there's a lot of different ways to get involved in research and labs on campus. I would encourage you to go to the Undergraduate Scholars Program, USP, and there's a lot more information in there. Okay, I think we're good, 9:50. If you have any more questions, just feel free to come up.